Again, good morning, grade 10. We are in the middle of the discussion about DNA and evolution. We are still in the process of how these genes are actually copied and transferred to the offspring. Okay, let's continue with. Can you see my shared screen?
Yes.
Yes.
Okay, let's continue with the copying the recipe. The recipe there is the genes or the genetic characteristics or the instructions in order for the ribosomes to create certain proteins. Okay, the recipe for cooking the protein needed by the body is only confined in the nucleus of the cell. The DNA nucleus also serves as the protective barrier to DNA. This DNA therefore must be copied because there are many molecules that can damage the DNA. One good example that can damage the DNA is the cancer cells. Number one, okay, the metastasis or metastasized um, cancer, it can spread all throughout the good cells. Another factor that can damage the DNA is to the radiation. Okay, those are factors that can damage the DNA in the cytoplasm and the machinery to make protein found in the cytoplasm as well, your ribosomes. Proteins are made up of chains of amino acid called polypeptides. Okay, poly means many. Okay, it is chains of amino acid. The transfer of information from DNA to protein is called gene expression. Okay, if it is actually transferred, the actual characteristic is being transferred, it can now be seen as a gene expression. It occurs in two steps. Number one is transcription or transcribe. Okay. Number two is translation. Transcription and translation. Let's tackle first transcription. Okay. Transcription, it actually writes genetic code in the DNA into the messenger RNA or mRNA. It works just like DNA replication and follows the base pairing principle except that the adenine in the DNA pairs with uracil. The one that I'm telling you last meeting, that adenine, instead of having uracil as a base pair, it is actually what? Replaced with uracil. What is the purpose of replacing um, thymine with uracil? Simply because this is now the transcription. The RNA or the messenger RNA cannot actually uh, carry the thymine because thymine is the finished product. It is actually one of the in order for us to create that um, certain protein, we need to have uracil, okay, to identify a particular protein. When transcription is finished, the DNA recoils to its double helix structure. The mRNA is now able to leave the nucleus by way of the pores in the nuclear membrane. Okay? It is called, the pores in the nuclear membrane is called nuclear pores. Okay? Katong mga lungag-lungag sa nuclear membrane. That is actually the passageway of mRNA. The mRNA carries the code to the waiting ribosomes in the cytoplasm. Okay? Ribosomes are the site of protein synthesis or the protein factory of the cell. The mRNA now, or the messenger RNA, possesses the code that specifies the order in which amino acids will be joined to form a polypeptide. Okay? The second part is the translation. Remember that now your mRNA is carrying the polypeptide. It possesses now the genetic characteristic and converted now thymine into uracil. So adenine is actually paired now with uracil. Okay? This is the second step in the gene expression whereby codons, consecutive three nitrogen bases, diba? Diba codons is contains three nitrogen bases or three pairs of bases, are
Yes.
read and translated into amino acids. Okay? Ribosomes consist of ribosomal RNA or rRNA. Okay? A functional ribosome consists of two subunits, a large and small subunits. The mRNA enters a ribosome. Let's take a look at what are these large and small subunits. Since the code in the mRNA does not have the punctuation mark that might signal the start and end of the codon, okay, it doesn't contain any. Okay, there must be a particular starting point, okay, or start here codon to begin with the translation process so that the whole sequence will be read correctly. Because without the starting and without the ending, it cannot be translated well. Remember that we need to start and we need to end. So what are these start here codon and the end codon? To begin the translation process so that the whole sequence will be read correctly. This codon is the initiation codon. Okay? It is called the initiation codon, the first AUG, okay, in the mRNA. AUG stands for again adenine, uracil, and guanine. In the messenger RNA or mRNA, that is also codes the amino acid to methionine or AUG. AUG is methionine. I will let you see later the table wherein you can actually see what the actual set of codons translated or shall we say the translated set of codons with regards to the particular protein being created by the ribosomes. The tRNA or the transfer RNA picks up the amino acid methionine according to their triplet bases or anticodon. Again, tRNA or transfer RNA picks up the amino acid methionine. Di ba methionine is your starting codon, right? Initiation codon to their triplet bases or anticodon. Since it is AUG, the anticodon for the start up or start now, start here, or the initiation codon is UAC. Okay? UAC, that is uracil, adenine, cytosine, attached to one end of the tRNA. The anticodon of tRNA, UAC, binds with AUG at the P site of the large subunit of ribosomes. The next mRNA codon is lined up at the A site. Okay, what is A and what is P? Okay, the sequence repeats in the polypeptide chain, grow until the ribosome reads any of the stop codons. These are the stop codons. Okay, please take note. We have uracil, adenine, adenine, uracil, guanine, adenine, or uracil, adenine, guanine. Are you familiar with these base pairs? We already discussed the base pairs, right?
Yes.
Okay. The ribosome gets stuck waiting for the tRNA that will never come and eventually falls off, releasing the new synthesized protein with the help of this special protein called the releasing factor that binds to stop codon and releases the mRNA or the messenger RNA from the ribosome. The mRNA may now bind with another ribosome for translation, okay, and may be broken down by enzymes. The polypeptide goes to the Golgi complex or Golgi apparatus or Golgi bodies to combine with fats, minerals, or carbohydrates to make complex proteins. Okay, we're talking about a lot of proteins here. We're talking about binding with the anticodon. Let's see if we can have the table of these proteins. Okay, this is now your table of those proteins. Okay, we have the first letter on your left side. On the upper part is the second letter, and the third part or the third letter is in your right side. Okay, you can see here the stop codons. It was actually identified there. The AUG or methionine is actually identified there. Okay, and of course all the proteins being used all throughout or being created all throughout the process. Okay, sure. That is the reason why thymine should be converted into uracil in order for the messenger RNA to carry this information. Okay, remember that the start button or the start codon is in AUG or the methionine, and the stop codons are UAA, uracil adenine adenine, uracil adenine guanine, and uracil guanine adenine. Okay. Question so far,
None so far.
What are those stop signs, sir?
meaning those are stop codons. Remember the stop codons that we are actually talking about a while ago? If we have an initiation codon or a start codon, we should have also stop codon. Because if we don't or if we will not have this stop codon, tendency is the translation will continue to translate until the end of um, the sequence of the DNA. It should be stopped. Why? Remember that over also of the creation of these proteins can lead to uh, death to a certain organism. Okay, upon studies, it shows that the creation of these proteins also has a positive and negative effect towards an organism. Okay, it could be a positive effect that we can create as a variety of certain organism in plants, but usually in animals it is actually fatal. Like for example, um, some genetic problems, born with the genetic problems, like um, a baby born without skull. Number one. Number two, a baby is born without arms and limbs. It could be beneficial, it could be fatal to an organism. Okay? How about a plant which is bigger than the usual? Okay? This is based on the genes. It could be beneficial for us. Okay? It could be beneficial also for the plants. Those are the factors. Okay? Upon checking, it should be incomplete or it should be completely um, lined up or sequence based on the actual genetic characteristic. Okay? A change of that sequence can lead also to certain death to some organisms. Any other question? Any other question? Grade 10?
None for me so far.
None.
Please turn on your camera, by the way, so that I can actually see you. Let's move forward. Anyway, this table is actually given in your module. It is in the module. You can download your module actually in the LMS. Mutation. Okay. We need to discuss mutation because this is a big factor also for an organism. Okay. Genes control the characteristics or traits that you inherit. If a change happens in the gene or chromosome, the traits will correspondingly be altered. Two, a permanent change in the gene or chromosome of a cell is called mutation. Okay, mutate.
Chair, excuse me, Chair.
Yes.
Is
Okay.
cancer considered as mutation?
It could be. It could be. But talking about cancer, to tell you honestly, I will just give you an information. We are all has cancer cells. That's apparently based on study. Why? Um, as human itself, it is included in our genes. I've studied genetics for a long period of time, and we've seen a lot of cancer cells in our own cells. It is called oncogenes. Or you can search about it. It is called oncogenes. Sure, why it is not manifested all throughout? It is not manifested because it is inactivated. It is inactivated. That is why I am giving you an idea about this because these oncogenes can be activated. How? Remember that the food that we take in, considering the fact that the environment is polluted already, can actually activate the oncogenes. Sure. Why do we have a lot of cancers to certain people exposed in like for example radiation or exposed in um, certain climatic conditions? These are the factors that can actually activate the cancer cells. Okay. I have also identified some of these carcinogens. It is called carcinogens. Number one is the soft drinks. It is one of the greatest carcinogen. It can alter your genes actually. Sure, why the soft drinks? Your soft drinks is composed of um, certain sugar. They otherwise called it as artificial sugar, magic sugar, or in medical it is called aspartame. Aspartame is one of the carcinogen, the greatest carcinogen, and it can really cause cancer. By the way, talking about a mutation, there are a lot of factors also to think about mutation. Like for example, remember the electromagnetic waves that are topic in the previous module.
Yes.
Okay.
Yes.
Remember the gamma rays,
Yeah, the strongest one.
Mm-hmm.
the strongest one, yes. It can actually create mutation. Okay? It can also, if it can cause, or if it can cure cancer, it can also cause cancer. Any other question before we move in?
None so
Right.
far.
Hawk is one of the experiment before, even though it is actually a, a character. Hawk is one of the experiment before with gamma radiation. Okay, and they actually let us identify or let us see how this radiation or gamma radiation acts on the body. It changes the body. Shall we move forward?
Sure thing.
All right. Point mutation is a mutation that exchanges one nucleotide for another. It may be harmless if the change is in the codon of the same amino acid. This is now the changes. So na exchange um, in same area, it actually changes or they exchanges places. So it creates now, or the nucleotide exchanges places. So it creates now a certain mutation. It is called point mutation. It may be harmless if the change is in the codon for the same amino acid, but it may also cause a change that may lead to disorder section of DNA. If the change happens in any of the body cell, the change may not be life-threatening. Like for example, extra fingers, okay, divided chains, dimples. These are mutations. What else? Um, Usually it's extra fingers. I've seen a lot of extra fingers. So it is not actually life-threatening. Okay, with um with a lot of bones.
What about like genetics, teacher?
Genetics. Yes, this is part of the genetics actually. The makeup. Okay. Those are just mutations. Like for example, also I have seen a a person which has a lot of teeth. Okay, he is actually featured um in most of the documentaries. Okay? Those are genetics. Okay, those are mutations. Any other question?
None so far.
However, if the change take place in the sperm or egg, this is fatal. The daughter cell from the division of these parent sex cells will altogether be affected by the changes or change, and the mutation will be passed on to its offspring. While many mutations are harmful, some are not. However, in extreme cases, mutations can cause death. Okay. Sure, what does it mean? Okay, some of the mutations are in the sperm or in the egg. Okay, you can actually pass it on from the parents to the offspring. Okay. Now, with this, it can actually affect the individual. Like for example, have you seen the documentary about um he actually thought or she actually thought that he is or she is a male because she has a sex organ or male sex organ. But then again, upon checking on her genetic characteristic, it shows that she is actually an XX or female. Okay? Those are just mutation. If it carries now, or if she is actually carrying that specific genes, she can pass it on to the next genome or to the next generation. And this can cause death. That is why in the medical field, they constructed genetic engineering. Okay, genetic engineering, the aim of the genetic engineering is to remove those mutations and replace it with a good genes. Okay, let's move forward. Any other question? Question so far?
None.
Okay. Biodiversity and evolution. Let's discuss biodiversity and evolution by next meeting. Do you have any questions so far with the previously discussed topic?
Sure.
Yes, scenario.
I don't know if this makes sense, but there are like transgender who wants to change physical traits of theirs, and they are taking medicines. So those medicines, do they contribute to the mutation of that person who wants to be on the other sex? Like he
That's
wants to be transgender.
Yes, of
Di ba
course,
naman sila git? Okay.
it can. Remember that we are actually driven by hormones. Those are hormone therapy. Okay, and changes of those is, take note, changes of those can lead also to some cancer. They're all carcinogen. Those are artificial. Remember that we are created naturally by God. If you're doing so with hormone therapy, I identified some of these hormone therapy, like Diane, oh, the injectables. I've seen a lot of those. And one of my students also, um, yes, Trisha, it could possibly lead to death. It is, especially if you have undergone overdose. Some of them are overdosing themselves. Why? Because they want the effects as soon as possible. Our selves or our body needs to adapt to certain changes. Okay, certain rises or certain fall or decrease of this level can cause some changes also in ourselves. Remember also that this can cause harm to your internal organs. That, for example, number one is your kidneys, number two is your liver. So those are affected. And hormones, these are not natural. These are artificial hormones. The purpose while um, bisexuals inject these hormones, estrogen, is they want to have a bigger chest and a bigger hips because these are the secondary sex characteristics. Okay, they can have that one, but the problem is these are not a natural hormones, and this can cause cancer. Again, I repeat, these can cause cancer. These are carcinogens. Okay. Any other question? Any other
No,
question?
teacher.
Even the singer Charis Pimpenko, she's actually taking testosterone. Okay. Have you seen the changes? It's really drastic. And
Yes.
even if she is taking testosterone, she still has the menstrual cycle, the menstrual period. It's still the same. She cannot remove that one because it's natural. She is taking artificial testosterone. It's the same. Any other question? Do you have any questions so far? Still have 2 minutes left. If you don't have question, I'll be ending this meeting. Let's continue by next meeting for the diversity. How diverse are the organism and how this diversity or the evolution of biodiversity affects some organisms. Okay, we are now moving towards how adaptation happens on these organisms because without adaptation it cannot continue um its generation or it cannot continue its species, right? I think that will go for today,
Yes.
class. Thank you for your participation, grade 10.
Thank you as well.
Thank you
Let's
as well.
continue by next meeting. Have a nice day, grade 10. Goodbye.
Likewise.
Goodbye.
Bye bye.